Hello everyone, welcome back to O-Level Oasis, your guide to CAIE exams. We're currently working through Chapter 11, Transport in Humans. Up to this point, we've already completed the first 3 units of the chapter, and now we're moving on to the last part, Unit 11.4, which focuses on blood. We've learned about the circulatory system, including the heart and the different types of blood vessels that work together to keep the system functioning. Now, it's time to explore what actually flows through this system. We'll look at the components of blood, what it's made of, and why each part is important. So, let's begin. The first specification we need to focus on is specification number 1. This requirement asks us to identify red blood cells as well as the 2 major types of white blood cells, lymphocytes and phagocytes. We should be able to recognize them as they appear under a light microscope on prepared slides, and also when shown in diagrams or photomicrographs. So, let's begin. To identify red and white blood cells, we first look at the key structural features. Red blood cells have a distinctive biconcave disc shape and do not contain a nucleus, which makes them easy to recognize. White blood cells, on the other hand, are generally round in shape and do contain a nucleus. Although there are many types of white blood cells, for this topic we focus specifically on lymphocytes and phagocytes. These cells, along with red blood cells, can be clearly observed when examining blood smears under a microscope. Here you can see how these different blood cells look and how their structures appear when viewed in diagrams or under a microscope. First, we have the red blood cells. These cells appear as small, round, disc-like structures. They have a biconcave shape, meaning they are flattened with a slight indentation in the center on both sides. This shape increases their surface area for exchanging oxygen efficiently. Mature red blood cells do not have a nucleus or other organelles, allowing more room for hemoglobin, the protein that carries oxygen throughout the body. Their main role is to transport oxygen to the tissues. Next, we move on to the white blood cells, which play a major role in defending the body against disease. The first type is the phagocyte. These are larger cells with an irregular outline. Their most distinctive feature is a multi-lobed nucleus, and their cytoplasm appears granular because it contains lysosomes that help digest foreign particles. Phagocytes protect the body by engulfing and breaking down bacteria, debris, and other harmful materials. The second type of white blood cell we focus on is the lymphocyte. These cells are generally smaller and have a very large, round nucleus that occupies most of the cell's interior. Only a thin ring of cytoplasm surrounds the nucleus. This structure supports their role in specific immune responses, such as producing antibodies and recognizing infected or abnormal cells. Lymphocytes are a type of white blood cell that play a crucial role in the body's immune defense. When viewed under a microscope, they can be identified by their very large, round nucleus, which fills most of the cell. Only a thin layer of clear, non-granular cytoplasm surrounds the nucleus, giving the cell a clean and simple appearance. Functionally, lymphocytes protect the body against infections caused by viruses and bacteria. They produce antibodies, Y-shaped proteins that attach specifically to antigens found on harmful pathogens. This response is highly specific, as each antibody is designed to target only one particular type of antigen. Lymphocytes also contribute to long-term immunity by remembering infections the body has encountered before. In terms of size, lymphocytes typically measure between 7 and 14 micrometers in diameter. Their most distinctive feature is their large, round nucleus, which makes them relatively easy to recognize under the microscope. Then we have phagocytes. Phagocytes are a type of white blood cell responsible for protecting the body by engulfing and breaking down harmful microbes. Their main role is to detect, ingest, and digest pathogens through a process called phagocytosis. During this process, they use powerful enzymes to break down the invading organisms. This is part of the body's non-specific immune response, meaning phagocytes respond to a wide range of pathogens rather than targeting one specific type. When observed under a microscope, phagocytes can be recognized by their multi-lobed nucleus and the granular cytoplasm. The lobed nucleus makes the cell more flexible, enabling it to squeeze between tissues and surround pathogens effectively. Phagocytes are generally larger than many other blood cells, with a diameter ranging from 10 to 20 micrometers. Their key identifying feature is the multi-lobed nucleus, which plays an important role in allowing the cell to move, bend, and wrap around microbes during phagocytosis. This is how phagocytosis works. Phagocytes defend the body by detecting, engulfing, and digesting harmful microbes. Their multi-lobed nucleus gives them the flexibility needed to move through tissues and wrap around invading pathogens. Using their sensitive cell surface membrane, phagocytes detect chemicals released by bacteria and are drawn toward the site of infection. Once a pathogen is detected, the phagocyte extends its membrane around it and engulfs it, enclosing the microbe inside a sac called the phagosome. Digestive enzymes from granules inside the cell are then released into the phagosome, breaking down and destroying the bacteria. The remaining soluble debris is eventually removed from the cell by exocytosis. This process is part of a non-specific immune response, meaning phagocytes respond to any harmful microbe rather than targeting one specific pathogen. As seen in the diagram, phagocytosis begins when a phagocyte encounters a bacterial pathogen, recognizing it as a foreign invader. During engulfment, the phagocyte's membrane wraps around the bacterium, pulling it inside the cell. The bacterium is then enclosed in a phagosome, where digestive enzymes are released to break it down. Finally, the bacterium is destroyed, effectively removing the threat and protecting the body. Now, if we look at this diagram, we can clearly see the different blood cells present, red blood cells as well as the two main types of white blood cells, lymphocytes and phagocytes. By observing their structural features, we can identify each one quite easily. The red blood cells, or erythrocytes, appear as numerous small, biconcave discs. They are the most abundant cells in the blood and can be recognized by the distinctive disc shape and the absence of a nucleus. Their primary function is to transport oxygen throughout the body. Next, we have the lymphocytes, which are a category of white blood cells. They lack visible granules, so they are classified as agranulocytes. In the image, they appear as large, purple cells with an almost completely filled interior due to their large, round nucleus. Only a thin ring of cytoplasm surrounds the nucleus. These cells play an important role in specific immune responses. Finally, we can identify the phagocytes in the diagram. These include neutrophils, eosinophils, basophils, and monocytes. Neutrophils, eosinophils, and basophils are granulocytes, meaning they contain granules in their cytoplasm and have a nucleus with multiple lobes. Monocytes, on the other hand, are agranulocytes and have a kidney-shaped nucleus. All of these cells function as phagocytes because they engulf and break down foreign particles. They stand out in the image due to their larger size compared to red blood cells and the distinctively shaped purple nuclei, whether lobed or curved. Also, we can also identify these blood cells in a photomicrograph, which provides a real microscopic view of a human blood sample. In the photomicrograph, the different types of cells are clearly visible and labeled for easy identification. The red blood cells are the most numerous, appearing as pink, biconcave discs without a nucleus. The phagocytes, which are a type of white blood cell, can be seen with the characteristic multi-lobed nuclei and granular cytoplasm, reflecting their role in engulfing and breaking down foreign particles. The lymphocyte, another type of white blood cell, is easily recognizable by its large, round nucleus that occupies almost the entire cell, leaving only a thin rim of cytoplasm. This visual representation reinforces how red and white blood cells differ in structure and function, making it easier to identify them in both diagrams and actual blood smears.